Good morning. Thank you to the organizer for the kind invitation to participate to this important webinar. It is my pleasure to be with such a distinguished panel of speakers and audience. In my presentation today on New Open Horizon for Dengue, I'm going to provide an overview of TAC-003 candidate vaccine clinical profile as a potential additional preventative tool for reducing dengue disease burden. Here are my disclaimers. The presentation is intended for healthcare professionals only, not for public, and the candidate vaccine discussed here has not yet been approved for any indication, and there is no guarantee that it will be approved for such use as discussed in this presentation today by any regulatory authorities. But let me start by describing the structure of this vaccine. So the vaccine is a live attenuated tetravalent vaccine containing a live attenuated dengue 2 virus strain in red and 3 recombinant dengue strains generated by replacing the gene coding for the envelope and pre-membrane proteins of dengue 2 attenuated virus with genes derived from dengue 1, dengue 3, and dengue 4, as shown in different color in the picture. The objective of this formulation was to activate a broad immune response against structural and non-structural components of the virus. This vaccine has undergone an extensive clinical development program, as you can see described here by looking just at phase 2 and phase 3 clinical trials performed. And you can see in different colors that this vaccine has been studied in children, adolescents, adults, in endemic and non-endemic countries, looking at immunogenicity, safety, and efficacy. The overview of this study has also shown that the activities, if we consider the full clinical development program, has been done in 18 clinical trials performed in 13 countries overall, and have involved more than 80,000 subjects, including seropositive and seronegative participants at baseline, aged from 1.5 to 60 years of age from endemic and non-endemic regions. TAC-003 was able to demonstrate that was able to activate multiple arms of the immune system, and the immune response was sustained against all 4 serotypes up to 4 years after the second dose. The 2 doses administered 3 months apart was the preferred schedule in terms of immune response. It was selected for the phase 3 pivotal studies I will discuss much more in detail. The candidate dengue vaccines also shown to be well tolerated with no important safety risks identified to date. So that's the overview, but let's move on now into the details of the pivotal phase 3 studies called TIDES that is still ongoing. Here is the content of what I will be presenting to you today. So I will focus on obviously on the study endpoints, the study design, and key results in terms of efficacy, immunogenicity, and safety. So starting from the TIDES endpoint and objectives, here are the details of all the endpoints, and I want you to focus the attention into the pyramid shown on the right, where you see the hierarchy of importance based on the statistical power of the study to detect the real effect of the candidate vaccine versus placebo. The primary endpoint in red was really to look at the ability of the vaccine to protect against virological confirmed fever induced by any dengue serotype at 12 months after the second dose. The secondary efficacy endpoint in blue was there again to look up to 18 months after the second dose, to look at the ability of the vaccine to protect against hospitalization by any serotypes, by baseline serostatus, seronegative and seropositive, by severity, so dengue hemorrhagic fever, severe dengue, and by serotype. The last but not least were the exploratory endpoint that you can see in green. The exploratory analysis obviously are not powered, but they are there just to inform future study hypotheses and for which the study obviously finding might be interpreted with caution, and will look in a series of different elements into efficacy by serotype, serostatus, by age, and so on. Obviously, along the trial, there was assessment of safety, and assessment of reactogenicity and immunogenicity in a subset of participants. So the study has been conducted and is still ongoing, as I said, in 26 sites across 8 countries. And the decision to go with such a large geography is really to cover different endemic regions of the world with different burden of diseases and circulating serotypes. The study design is quite complex. The study enrolled more than 20,000 children aged 4 to 16 years of age. The target age was selected to make sure enough proportion of seronegative subjects could be included, and considering also the age range with the highest risk of symptomatic severe dengue in the countries involved. The study participants received 2 doses 3 months apart of the TAC-003 or placebo in a 2-to-1 randomization ratio. An active and modified surveillance system has been followed throughout the trial, with participants or their guardians contacted at least weekly to ensure identification of dengue cases. And the study follow-up is quite extensive and has been planned in part, again, to assess the primary endpoint, the secondary endpoint, and then the long-term assessment into the exploratory endpoint. A booster study is also being launched at the end of the phase 3 trial in order to assess again the added value of a booster dose for such a vaccine. So moving on into the study population and demographic of the study recruitment, we can see here that there was obviously a very comparable split in terms of subject enrolled and characteristics. You see that about 27% of seronegative subjects were equally distributed among the placebo and vaccinated cohort. So this is important because the study really can allow us to assess the ability of inducing vaccine efficacy based on baseline serostatus. It was also, there was a good split between different ages, with 12% of subjects that were in the younger age category, so a slightly smaller number of subjects there, and 45% were from Asia versus 55% from Latin America. So the efficacy data that have been obviously reported first because they were calculated up to 12 and 18 months respectively are summarized in the slide I'm showing you now. So let's start with the primary endpoint up to 12 months after second dose, and here the results. So TAC-003 demonstrated 8.2% efficacy against symptomatic virological confirmed dengue fever up to 12 months. The primary endpoint of the study, that was exactly that per protocol, was met, and all 4 serotypes were reported, but very little presence of dengue 4 was there, as you can see on the image on the right-hand side. These data from a clinical perspective are important because they might give an indication of possible reduction in number of outpatient dengue fever cases that we might observe. Moving on to the secondary endpoint, again, this time looking up to 18 months post-dose 2, here the results in terms of hospitalization rate. TAC-003 demonstrated 9.4% efficacy against hospitalization caused by virological confirmed dengue fever. And the secondary endpoint, again, in this case for hospitalization, was met. And this is important because the hospitalization parameter gives an indication of the possible reduction in the number of inpatient dengue fever cases, potentially reducing the bed occupancy, burden and cost on the healthcare system. So that's also quite an important element to consider. Moving on into the severe cases, TAC-003, you see here the split of the cases observed, demonstrated 85.9% efficacy against dengue hemorrhagic fever up to 18 months, and according to the WHO 97 criteria. However, we had also dengue case adjudication committee that was an independent group of experts assessing the cases in a very objective manner, and they identified very few severe cases based on their criteria, and this did not allow for a statistical significant evaluation of the 2 groups. As you can see here, we have very large confidence interval. Moving into the efficacy by dengue serotypes, here the results, we see that obviously the vaccine was able to demonstrate a high efficacy for dengue 2, followed by dengue 1 and dengue 3. And the secondary endpoint were met for these three serotypes. However, the vaccine efficacy against dengue 4 could not be statistically evaluated due to the insufficient number of cases. And as you can see, very large confidence interval, including the 0. Moving on into the efficacy by baseline serostatus, this is the overall results, again, up to 18 months. TAC-003 demonstrated efficacy against symptomatic virological confirmed dengue, regardless of baseline serostatus, with 73.3% overall, with 76.1% in seropositive and 66.2% in seronegative. So point estimate slightly lower for seronegative, but overlapping confidence interval. Moving on into the hospitalization, again, a similar example as before into the overall cases. We see a better picture because we see a higher protection against hospitalization in dengue seropositive, 91.4, and a slightly lower 88.1 in seronegative, but still with very overlapping confidence intervals. So it means that the efficacy is comparable between the two groups. Continuing the observation by serostatus and serotypes, here is the overall assessment up to 18 months. So TAC-003 demonstrated variable efficacy at this point, and the variable efficacy was really linked to the different serotypes. For sure, the vaccine efficacy in seropositive, seronegative was established for dengue 2 and dengue 1, and was established in dengue 3 seropositive. But there was no efficacy observed in dengue 3 seronegative with very large confidence interval, including 0. And in dengue 4, there were very few number of cases reported that did not allow for a statistical significant assessment. And let's move on now into the exploratory follow-up data up to 36 months, where we look again at efficacy, immunogenicity, and safety. So up to 36 months, that's where we are in terms of the study published findings so far, and that's what I will be presenting to you now. So here is the overview overall in terms of efficacy against symptomatic virological confirmed dengue fever. And here you see the overall seronegative and seropositive. So we see a tendency towards decline between as compared to the 18 months and the 12 months I showed you before, but still with protective efficacy that is quite sustained. At the same time, what is quite interesting on the right-hand side of this slide is really the Kaplan-Meier curve that shows really the percentage of subjects with cases into the placebo, that are the thick lines, seropositive, seronegative, versus the vaccine group that are in the dotted line. And again, what is important to see is that there is a sustained separation of the two curves already starting from the first dose, because there is the first separation already between 0 and 3 months. That's quite important and gives the idea that the population overall might benefit for such a vaccine regardless of baseline serostatus. And if we look in terms of the number of cases that could be prevented per 100,000 vaccinated subjects, this will be more than 4,600 in seropositive and 3,900 in baseline seronegatives. Moving on into looking into this hospitalization, again, the hospitalization looks and remains quite high sustained. So the good news is while there is a decline, this decline seems to really be linked to more outpatient subjects, while for hospitalized dengue fever, the decline is not really there because it's quite sustained. We had 90 before, we have 86, 83 now. And still, again, overlapping confidence interval and a good picture for both seropositive, seronegative. And when we look again on the right on the Kaplan-Meier curves, we see again this very strong separation between the vaccinees and control, showing again a possible benefit of this vaccine. In fact, per 100,000 vaccinated subjects, that we could prevent up to 1,600 cases in baseline seronegative or up to 1,400 cases in baseline seronegative. Seronegative. Moving on again into the exploratory analysis up to 36 months against severe dengue and dengue hemorrhagic fever, we see dengue hemorrhagic fever efficacy was 65.4, again based on the WHO 97 criteria. At the same time, obviously, once again, the efficacy against the severe dengue based on the dengue case adjudication committee group was again still not statistically significant because we had very few cases reported overall. Moving on now into the immunogenicity that was observed in the immunogenicity subset in this study. Here we look at the immunogenicity in the subject that were seronegatives at baseline. And as you can see here, is that the graph represents the geometric mean titers reported in a logarithmic scales in individuals receiving the candidate dengue vaccine in thick line per serotype versus the individual receiving the placebo. These are the dotted line. So GMTs remained well above the baseline titers, especially for dengue 2, and stabilized around 9 months because they had the peak and then the decline, with 80.5% of subjects remaining seropositive against all 4 serotypes up to 36 months of follow-up. If we look at the same picture into immunogenicity in baseline seropositive, obviously the difference between the two is less evident because they had already antibodies. But what is very well shown here is that the GMTs for all 4 serotypes increased after TAC-003 and remained higher than placebo, with the similar pattern of stabilization from the 9 month onwards up to 36 months of follow-up. If we move now on into the safety profile, we start from the tolerability, so the local safety, and here again the graphs on the left-hand side shows the injection site reaction up to 7 days after any vaccination, and on the right-hand side we show the solicited systemic adverse events up to 14 days, divided again for young subject versus a slightly older subject. The solicited local reaction, the most frequently reported one were injection site pain, followed by systemic adverse events like headache, myalgia, malaise, and asthenia. But overall, the picture looks very reassuring and very similar in terms of profile between vaccinees and control. The big difference was really into pain at the injection site between the two groups. When we go into the total rate of unsolicited serious adverse events explained here in this table, we can see, again, by looking first of all at any and then the most frequently reported ones, that the rates of unsolicited adverse events were similar in the vaccine and placebo groups in terms of frequency. The most frequent serious adverse event was infection and infestation, but this was uncommon, in less than 4% in either arm of the study. And of those, mainly the reports were nasopharyngitis, upper respiratory tract infection, and viral infection. Moving on into what is the most important element that everybody is interested upon is really the severe adverse events reported over the study up to 36 months of observation or follow-up. As the data are showing here, there were no clinically meaningful differences in severe adverse events between the experimental and placebo arm overall and by serostatus, and that's extremely important. No severe adverse events were judged to be vaccine-related, including reported deaths. Overall, no important safety risks were identified up to 36 months of follow-up after the second dose, supporting the good tolerability and favorable safety profile of this candidate vaccine. Most importantly, there were no signs of possible enhancement of disease. And the last slide around safety, here is really looking at the frequency of severe adverse events, and you can see that they were similar across placebo and vaccine group. I remind everybody that this is 2-to-1 randomization rates. And here, as you can see, the list of most frequently reported one, there was no clinical meaningful difference between the experimental and placebo arm. And again, there does not appear to be an increased risk of severe diseases or hospitalization post-infection when vaccinated with TAC-003 for individuals seronegative or seropositive at baseline. And that's, again, a very important finding. So this brings me to the conclusion slides, where I summarize the data I showed you, because there were quite a few in a row. And here the key element I want to say is TAC-03 really demonstrated overall efficacy against symptomatic dengue in both baseline seronegative and baseline seropositive children aged 4 to 16 years. This was the efficacy for the primary endpoint, and here is the summary of the efficacy I presented to you for the secondary endpoint. The exploratory analysis in the long-term follow-up, we observed a tendency to decline in vaccine efficacy in outpatient subjects, as it is observed in other vaccines, but still a robust efficacy that remains up to 83.6% against hospitalized virologically confirmed dengue, regardless of serostatus at baseline. We have seen as well variable efficacy against serotypes, in particular lack of efficacy against dengue 3 in seronegative individuals, and very few dengue 4 cases that will require further assessment. Last but not least, the reassuring element of no important safety risks identified up to 36 months after the second dose, and most importantly, no signs of possible enhancement of disease. So these are overall the data observed in children and adolescents, 4 to 16 years of age. And today, obviously, the question might be the generalizability of this finding to young adults and adults in general for countries where they see cases in these age categories. And to answer this question, what has been done in the clinical development plan of this vaccine, what we have done, we have performed what are called immunobridging assessment to link efficacy and immunogenicity of this pivotal trial with other trials in adults by showing that there is non-inferiority in the immune response between the what's observed in this efficacy study versus what observed in adult study. This data will be published in the coming months and have been submitted to regulatory authorities to support the indication from 4 to 60 years of age that we are seeking. In conclusion, the TAC-03 clinical benefit risk profile is favorable and is encouraging to consider this candidate vaccine as a potential additional tool that brings hope in the horizon to help reduce the burden of dengue diseases on individuals and on the healthcare system. And with that, I thank you for your attention.